Swayam Prabha. Digital India. Educated India.
Welcome back to the organ printing course. In the last few lectures, we have discussed on about the bioinks and their role in bioprinting, and also what are the different characteristics of bioinks, what are the different bioink properties, and so on. In this lecture, we will discuss another important point or another important component of bioink, that is single material and multi-material bioink. If you see in literature, there are you can find either some of the bioink of made up of single material and some other bioinks that can be made up of multi-material. So we'll see in this lecture what are the different types of what is single material bioink and what are the limitation of a single material bioink and how a multi-material bioink can be developed and what are the different ways or applications of this multi-material bioink. As you must have understood, that single material bioink systems are mostly made up of single polymer, where a single polymer like gelatin, alginate, fibrin, collagen, or any other single polymer, or even synthetic polymer like PVA or any other thing, can be dissolved in a solution in water, and then or in buffer or media, and then that polymer solution hydrogel can be used as a bioink. So that is called single material bioink system. And as the name suggests, because of this single material system, it is easy to use. Okay, so that single material bioink system is most user friendly. It is the manufacturing of this thing of the or the processing of this bioink, preparing this bioink, that is also very easy. Also the user, it is very easy to use, right? And also you can, with single material bioink system, you can design straightforward experiments. That means when you are trying to use the material for bioprinting, single material system can give you better design control. Like you can design the experiments very well, and you can perform the experiments very so it is most straightforward because the single material system, so you can control the concentration of the polymer in that solution, by which you can also control the viscosity of the material in the solution, and you can bioprint with single material system. Other thing is, when we are working with single material system, then it is very easy to, or is very easy to understand the cell material interaction. That means when the material cells are incorporated within the bioink, that cells, how the cells respond to this the material, to interact with the material, respond to the material, how the cellular proliferation, cellular migration, differentiation, if it is themselves, and so on, so on. So all these things can be very easy to understand, can be very easy to study, right? Because there is a single material, so whatever the effect, whatever the influence that can have on the cells, that is because of the single material. So it is very easy to study. The other thing is that because of this single material, because it is composed of single material, it is very easy to prepare and process. So that means the minimizes precursor processing and preparation. So the preparation procedure of single material bioink system is very easy, because you can very well make that bioink just by mixing one polymer or hydrogel in a particular solvent, and then you can directly use it for bioink. And the other thing is, when you look for translation of this your bioink system for biomedical therapies, suppose when in the future, suppose that one bioink is used for making some 3D structure, and that can be translatable, so that if it is a single material system, this helps enormously for translational application, because many a times the regulatory board, there are regulatory challenges, but that demands a stringent manufacturing procedure with quality control procedure is required to get regulatory approval. So whenever you want to translate, one wants to translate a bioprinted product for clinical applications, then there are stringent requirements for regulatory approval. So that stringent requirement can be easily met if it is a single material bioink system, because you need to take care of that. It is very easy to take care of single material than multi-material, because if there are multi-material, then there are various combinations possible, and each of these combinations have their own properties and challenges. But with single material, that is not there, and that's why single material bioink system is very easy to use, straightforward, easy to study the cell material interaction, and also it facilitates future translation easily. And if for regulatory approval, the meeting stringent manufacturing quality control procedures is easier. But with single material bioink system, there are some limitations. Because of its single material nature, there is a in certain cases it lacks the printability with inadequate post-printing mechanical properties, because we can make structures, we can print with single material system, but as it is made up of single material, so the viscosity regime of that material can be changed by changing concentration of the polymer. Suppose changing concentration, changing molecular weight, as in the last seg lecture we have discussed all this. So lack of printability with inadequate post-printing mechanical properties. The mechanical properties of the printed structure many a times are not great. So with that, there is a challenge. Then with single material system, you cannot print a structure with high print fidelity and shape, and where the shape can also be written. That is very challenging with single material. Because of this, the printability of the material is such that you cannot generate a structure with very high print fidelity and the shape retention. One way to tackle these things, to improve the printability, also to improve the mechanical properties, and to enhance the shape retention, is increasing the hydrogel polymer concentration. If you increase the polymer concentration, if the viscosity of the bioink goes high, and due to that high viscosity, it helps in better printability. So the printability can be improved by increasing polymer concentration. But please remember, as I have discussed in the last lecture, cells prefer high permissive environment, or say softer microenvironment is required for cells to migrate, to deposit the matrix, to proliferate, to do various cellular functions. Cells prefer high permissive microenvironment. Right? So for that, just by increasing the polymer concentration to improve better printability, that you cannot achieve the functionality, cellular functionality can be reduced. So if we increase polymer concentration just to improve better printability, then that structure lacks biofunctionality, so that the cell viability and cell instructive capacity, those things will be lacking in those, because within that dense microenvironment, as I have discussed in the last lecture, that if you increase the viscosity, the polymer density can also increase, and then that the polymer mesh network can also increase. So all this leads to increase mechanical property. That is okay, that is good, that can be good for better printability, but all this also leads to lower cellular responses. So cellular functions, cellular responses would be low in that case of things. So it results in less permissive hydrogel microenvironment. Due to that, the cellular response, cellular functions can be decreased because of this thing. That's why with single material Bioink system, we have a very narrow biofabrication window, because we cannot use very low viscous Bioink. When the viscosity low, the concentration of the, suppose when the concentration of the material is low in the ink, the viscosity is also low, then it is not good for it is not good for printing. The print fidelity is not great, but probably that is very good for cell viability or cellular functions. But on the other hand, when if you increase the viscosity again to very high extent, then what will happen? If you increase the polymer concentration to a very high, then the viscosity will improve, viscosity will increase, and due to the high viscosity, probably your printability will also improve. Your print fidelity or post-print shape retention, mechanical properties can improve further, but that kind of microenvironment, a stiffer microenvironment, is not great for cells. So we have a very small window, narrow biofabrication window, where we can work with but some low to moderate viscous Bioink or concentrated Bioink. That is good for cellular responses or cellular function. But spectrum of mechanical properties that is found generally in native architectures cannot be recapitulated by single material bioprinting system. Because of this, because of this, we cannot tune the material in such a way so that we can both printability, shape retention, mechanical property, everything can be achieved just by single material Bioink system. So the take home message from this slide is that we need a Bioink where maybe that material is good for printing, the printability is good, the post-print fidelity or mechanical property is also good, but it provides also a less stiff or less a permissive hydrogel microenvironment that is good for cell viability. So multimaterial Bioink systems are developed where it is made up of more than one polymer or hydrogel networks. So many times 2 or 3 or 4 different types of polymers or hydrogel networks are either mixed together or printed simultaneously in a single construct, so that we can address the limitations that we have seen in the earlier slide. So here, and another important property of this multimaterial Bioink system is, because there are more than one type of polymer chains or hydrogel network present, so we can also work on distinctly different gelation mechanisms for different components in that Bioink. Suppose the Bioink is made up of 2 different components, 2 different hydrogel networks. Then each of these hydrogel network can be solidified or gelated with one type of mechanism, and the other one can be used on them. Suppose these two hydrogel network, they don't interact with each other, they are just physically blended together, then we can work on those 2 different distinctly different gelation mechanism for a single construct. So in that way, we can improve the print fidelity, the mechanical post-printing mechanical properties, all these things by introducing distinctly different gelation mechanisms. Other thing is things when if you suppose if you have a buy you have a your bioink is made up of one component. If you include another component or secondary component to that, and if that secondary component brings a few biofunctionality, then that will improve the biofunctionality of the whole bioink, right? So that kind of things, like suppose addition of some cell bioactive or cell instructive components within a hydrogel improves the biofunctionality of that particular bioink system. So multifunctional bioinks, that is another major advantage of where we the improved bioactive multimaterial bioink system can be developed. Another thing is when you have one type of component, and if you are when you add another component to it, you can very well fine tune the rheological properties of this bioink, and by this way you can improve the printability of that bioink, and also you can improve the mechanical properties of this bioink, and so that the mechanical stability can be enhanced further of that resultant hydrogel of multimaterial component, and long-term shape fidelity is very much possible where the material will retain its shape for long term, maintain the shape long term shape maintenance can be possible. Other thing is you also have more control over the material properties of the scaffold 3D background, so that the scaffold's 3D background can also be controlled or can also be modulate or tuned with multimaterial system, because you have a control to change the proportion or the concentration of each of these hydrogel, the proportion of each of these hydrogel in the overall mixture, so that you can tune the rheological properties. The bioactive properties can be improved. And the other things also, you can get heterogeneous mechanical properties and biological features. So with multimaterial system, we can have heterogeneous mechanical properties. That means the scaffold's mechanical property can be varied at different zones. Suppose if we use two different multi-bioink component, two different hydrogels in a single construct, then we can deposit this bioink simultaneously at different locations, and we can control the mechanical properties or modulate the mechanical properties at different locations or different zones of that construct. By this way, we can get heterogeneous mechanical property, and we can get actually, because all if you see most of our tissue is the biology has the biological it's the biological nature of the tissue is that the tissue has heterogeneous structure, heterogeneous mechanical property, heterogeneous function. So all this can be achieved with multimaterial bioink system. That's why multimaterial bioink system is very important, and there are recently there is a huge interest in this domain. Well, there are various scientists working on to develop multimaterial bioink system. Now we'll discuss what are the different ways to generate multimaterial bioink system. Should we just mix the different components together and then develop the mixture? One way to do that is physically mixing that the components of the bioink in a together in the final form, where that can be used for the bioink. But the other way also, the first point is mentioned physical manipulation of bioink. Well, either the multimaterial bioink or the bioink can be patterned. That means at certain locations one type of biomaterial is printed, and another location other type of biomaterials are printed. So by this way we can basically pattern the different bioinks at different locations, and we can have different types of properties, different types of properties, and by this way we can generate different biological functions. Right? Physical mixing, as I said, also another way of doing that. The next is chemical manipulation of bioinks. Chemical manipulation of bioinks is mainly because it is chemical conjugation and cross-linking of different bioink components. Like we use, suppose we use more than one type of bioink or hydrogels or chemical polymer solids, polymer together, then we can have certain chemical reaction in between the components, so that these are like either chemical conjugation or cross-linking can be there, so that the bioink can be further stabilized and bioink can be further work. This chemical cross-linking, other things can also be done post printing or after deposition, after fabrication of the structure can also be used. This chemical cross linking can be used to further stabilization of the structure or to further improve the mechanical properties. So these are two ways, like either by physically manipulating the bioink, also we can develop a multimaterial bioink. Chemically also manipulating the bioink, we can develop multimaterial bioink. There are other types of bioink, those are also very, or those are also gaining popularity, or because of their inherent advantages of this kind of bioink, like nanocomposite bioink. Nanocomposite bioink, when in the, suppose in the hydrogel system or in the polymer solution, we incorporate nanostructures, whether it can nanoparticles, nanofibers, nanocapsules, or different nanostructures. When the different nanostructures are incorporated within a hydrogel system, then that composite is a nanocomposite, and that can be very well used for different bioprinting applications, because it has got huge advantages over a conventional bioink, conventional single system bioink, a single material bioink. Then they say one such example is when, suppose we incorporate this nanohydroxyapatite within this PEG DA, at PEG MADA. So in this polymer, polyethylene glycol diacrylate, when in this polymer, when we incorporate this nanohydroxyapatite, then the nanohydroxyapatite is the nanophase here, that is deposited or that is incorporated within this polymer, in this polymer system, then we developed a nanocomposite bioink. Nanocomposite bioink, we'll see in the next slide what are the different applications of this nanocomposite bioink. The other thing is the supramolecular bioink. Supramolecular bioink is the thing when the when more than one component or two components, like two different hydrogel networks, are in mixed or combined together, there is no chemical bonding or covalent bonding between these things, but there is a possibility of generating hydrogen bonding or ionic interactions, hydrogen bonding, or maybe some other kind of interactions present within the supramolecular bioink. So where both the different components can be cross-linked or cross-linked separately and can be used. Like the example is nanosilicate clay, alginate, and MC. When these things are mixed together and developed a bioink, they all have their own hydrogel network, but they can be mixed together, and then the supramolecular bioink can be developed, where some kind of interaction between the different components can be possible, and then be they can be used as a bioink for making different architectures. Supramolecular bioink and nanocomposite bioink provides different advantages, because by doing this, the mechanical property can be improved, the bioactivity can be improved further. Similarly, the printing fidelity can be improved, the the shape retention can be improved. So all these disadvantages, whatever the limitations we have come across for single material bioink, can be resolved, and the properties of this bioink can be improved with this kind of compositions. Now let us discuss what are the different applications, or what are the different characteristics, or what are the different applications of this nanocomposite bioink. Polymer nanocomposite bioinks are such where multifunctional nanocomposites incorporating nanomaterials such as ceramic nanoparticles, carbon nanomaterials, magnetic nanoparticles. So all these different types of nanoparticles where are incorporated within the biomaterial system, biomaterial or hydrogel system, and then a nanocomposite, okay, or a nanocomposite is developed. So that nanocomposite is a multifunctional one where it can function differently. Suppose so different properties can be improved further. So one such advantage of incorporating nanomaterials within the hydrogel or within the biomaterial system is the nanoscale surface topology can be improved. So we can have, suppose a biomaterial, we have a hydrogel biomaterial, hydrogel bioink, by which we can make a structures. And now where if we suppose also incorporate nanoparticles or nanomaterials, that nanomaterials can improve the surface topology. So it will provide some nano surface roughness can be created, and it has been proven or it has been observed that the cellular responses over this surface topology, the surface roughness, if the surface roughness increase, the cellular responses is far superior than the without this nano topology. So that's why. And the other thing is also this nano surface topology that induces cellular proliferation, cell migration, and also there are few studies where it has been proven that even the stem cells can also be differentiated just by doing this different surface, different types of nanostructures. If suppose there is a different nanostructures present on the surface, so that lead to differentiation of stem cells. So nanoscale surface topology is very encouraging to generate for on bioprinted structure, so that the bioprinted structures can also be as you can work as a cell instructive material or cell instructive structure where the cellular responses would be enhanced further. Similarly, by incorporating nanostructures within the hydrogel system, the bioactivity can be improved further, because the material will function better if this, because of this presence of these nanostructures within the material. Suppose the for example, when nanohydroxyapatite is a bioactive component. Suppose this nanohydroxyapatite is incorporated within the hydrogel network, and a nanocomposite is developed. So that nanohydroxyapatite nanocomposite material will function very well. So that helps in better bone tissue regeneration, even osteoinduction, because of this osteoinductive properties of this nanohydroxyapatite. That structure or bioprinted structure will have higher amount of osteoinduction inductive properties, so that it will be very good for bone tissue engineering applications. Similarly, by including similarly either as a bioactive component, either hydroxyapatite or bioglass, so can be incorporated within this thing, and the bioactivity can be enhanced further. Other thing is nanocomposite material also helps in. Nanocomposite material has higher mechanical property than the only hydrogel because this nanoparticle material, something like some nanoparticle materials like different types of carbon nanomaterials. When these carbon nanomaterials or graphene, when these are incorporated within the hydrogel material, the mechanical property improves further because of the arrangement of those nano materials within the structure present or within the bioprinted structures. The other thing is the conductivity of the structure can be improved further for certain innervated tissue engineering, because for innervated tissue engineering, though they need conductive materials so that the tissue building can be improved further. So conductive nanomaterials like carbon nanomaterials, graphene nanomaterials, all these different types of nanomaterial can be incorporated within a polymer matrix or hydrogel matrices, and then it can be used to prepare fabricate 3D structures. So that 3D structure will definitely have high amount of conductivity, and that will be very good for different innervated tissue engineering, like for heart tissue, myocardial tissue engineering, for nervous tissue engineering. This kind of nano conductive nanomaterials incorporated polymer matrices or nanocomposite bioink can be very well used. Other way to improve the functionality is just by incorporating magnetic nanoparticles within polymer nanomatrices. When magnetic nanoparticles are incorporated within the polymer matrix, that help that can provide that make the structure responsive to magnetic field. So the magnetism of the can be improved further, and by now when these magnetic nanoparticles are incorporated, then they can be also arranged by an external magnet. When you place it close to that, this magnetic nanoparticle can be arranged in a certain way, and by doing that we can generate different gradients of uh, different gradients can be actually produced within the structure, and by this way it helps in better tissue regeneration. The other property is the for photothermal effects also nanomaterials can be used. Suppose some nanomaterials, they are responsive to, they are as if they act as a photothermal, they have the photothermal behavior. Suppose when the light is exposed to these nanomaterials, they they absorb that laser energy, light energy, and converts them into a heat. So the material, they increase the heat of that system, or the heat of the system improve further, and because of this we can control or we can modulate the release of certain factors within that structure, so that can lead to better tissue regeneration. Other than this photothermal effect can also be used for killing cancer cells. So where suppose we incorporate these nanomaterials within a bioink, and the structure is made, and it can be used for treatment as a for cancer treatment. So when within the, suppose then it can be further, it can be, suppose it can be put into the cancerous tissue zone, and then when it is hit again, the light is on, then it can eliminate the cancer tissue from this thing used by the photothermal effects. So all these different types of examples, like different types of functions can be possible because of this nanocomposite bioinks. In this slide, I have listed few of the most commonly used bioink materials, both single and multi-component bioink material. So we'll see one by one what are the different types of, but this list is not exhaustive. There are many more materials available or present that can that is used, or those are used as bioinks for different applications. But here these are most commonly used, like alginate is the most commonly used, widely used material as a bioink material, because alginate can be cross-linked by introducing calcium ions to this material, and then the ionic interaction, ionic cross-linking happens, and due to this ionic cross-linking, that is also very for rapid gelation occurs, and due to that's why alginate is a very popular material for different applications. Other than this thing, alginate can also be used for encapsulating cells, and so that the for it is also you can be used as a bioink for when the silicon hydrogel for bioprinting. But as we have discussed earlier, only single material has certain limitations, so that's why alginate has been further combined with different other hydrogel materials to improve the property of these things. One drawback of alginate material is it doesn't have any cell recognition site, so cellular attachment to this bioink is very poor. Cells are mostly encamped, cells are just embedded within the alginate gel, they don't get attached to the alginate material. So there are different bioactive materials like collagen type 1, fibrin, gelatin, all these things are all these things are combined with alginate to develop multimaterial composite bioink, multimaterial bioink. Like alginate has been combined with collagen type 1, so the solar response improves further. Alginate also combine fibrin, gelatin, all these things. And then the cellular response is far superior than the only alginate bioink. Similarly, collagen type 1 has also been used as a bioink material, but the collagen type 1 material, it is thermosensitive material. The temperature has to be controlled for this thing material. There also collagen type 1 has been incorporated mixed with other biomaterials like fibrinogen to develop a composite, to develop a multimaterial bioink. Similarly, polyethylene glycol dimethacrylate, PEGDA. So that this is also a very good material for photosensitive material, or that can be photo cross linking can be used for this bioink, and by this way it can be used for any photo based any laser based or light based 3D printing techniques. Agar is another material that has been used as a bioink material. Agarose is also been used as a biomaterial bioink, as a material for bioink preparation. Collagen type 1, that is also used as a biomaterial bioink. Similarly, gelatin has been also a popular bioink because gelatin is inexpensive. It is the denatured form of collagen, and it can be very well prepared for different sources. So gelatin is also a very popular material. But gelatin, another thing is gelatin can work as a thermoreversible material, as I have earlier also mentioned. Below 30 degrees centigrade behaves like a gel material, and above that it behaves like a fluid material. So gelatin also has been used for different other applications. Similarly, gelatin methacrylamide another material where this can be used for, and gelatin methacrylamide where gelma, so the methacrylamide group is attached to the gelatin, and it has become photosensitive material, so it can be used for photo cross linking. It can be photo cross linked, and it can be used for different photo based, light based 3D printing technique, bioprinting techniques. Gelatin has been further some other material like alginate, chitosan, fibrinogen. All these material, or only chitosan material, all these materials have been also incorporated with gelatin to develop the multimaterial bioink system. Similarly, fibrinogen, hyaluronan, all these also incorporated with gelatin to improve the. One disadvantage of only using gelatin is this material, the viscosity of the material cannot be increased very high to improve the print fidelity, because the cellular response would be not that great. With highly concentrated gelatin doesn't provide a uh, very good microenvironment for the cell to serve cellular viability, cellular function. So that's why some other biomaterial has been incorporated, by which the printability can be improved further without actually increasing the density or concentration of the gelatin material. So gelatin methacrylate has also incorporated with hyaluronic acid. Also with gelen. Gelen gum is also another material which acts as a rheological modifier, by which the rheology of the material can be improved, printability can be improved without adding too much material into the system, or so that the cellular viability, cellular function need not to be compromised. Hyaluronic acid has also been used as a single material, and but it also has been used with other material like hydroxymethyl methacrylate and derivatives. All this material can be mixed together to come up with a multimaterial bioactive system. Similarly, hyaluronic acid methacrylate HAMA, where methacrylate group is also attached with hyaluronic acid, chemically modified. So that works as a photo cross linkable biomaterial, biomaterial. Gelatin methacrylate is another photo cross linkable material. Hyaluronic acid methacrylate has also been used as a single material for bioinking. Similarly, Lutrol F127, Matrigel, Methazole, all these different single biopolymer system has been used as a bioink. And there are other things like polyethylene glycol diacrylate, PEGDA, or in isopropyl mid and propylene glycol. Both these can mix together to develop this hydrogen. So these are some examples of bioink, different biomaterial systems where mostly they use as a bioinks. So there are many such bioinks materials, both single and multi material bioink systems available. Please, I encourage you to go through literature for more and more examples of this bioink material. Thank you all for attending this lecture. We'll see you again in the next lecture. Thank you very much.